Dr. Ripke here with Genetics Number 6, Part 1 of Protein Synthesis. Be able to characterize the steps of protein synthesis from DNA to proteins. This process is kind of like learning how to read. First, you learned how to make the letters. Then you learned how to put the letters together and start reading words. And that's what happens with DNA to proteins as well. Proteins, remember, are long chains of amino acids. There are 20 different types of amino acids, and these proteins might have anywhere from 50 to hundreds of different amino acids to make one protein. The order of the amino acids and the type influence the shape and function of the protein. Proteins include enzymes, insulin, muscles, and all the working parts of cells. The ribosomes make the proteins based on your genetic code order of A, T, Cs, and Gs. To build a protein molecule, the genetic information of DNA must get outside of the nucleus to the ribosome in the cytoplasm, or the rough ER. That DNA information is first copied into messenger RNA, mRNA. We call this process transcription. Transcription is when DNA information is transcribed into a mRNA molecule. The mRNA has the same information as the original DNA molecule, except uracil replaces the nitrogenous base thymine. The base pairing occurs as A goes with U and G goes with T. The mRNA strand separates from DNA and the DNA helix closes, and then the mRNA can leave the nucleus. We see that here in two pictures. Here's DNA. Enzymes open it up, an RNA copy is made along one strand, that RNA is released, and the DNA closes up. We could also see it here in this diagram. Here's DNA. Remember, all Tygos can growl. A goes with T, G goes with C. But when you add RNA, there is no T, so it becomes AU. So A goes with T goes with A, AU, C, G, etc. all along. The DNA opens up, RNA forms, they separate. That transcript of RNA goes to the ribosome, which eventually becomes different amino acids like methionine, isoleucine, and serine.